Assalamu alaikum students. In this lesson I will describe about artificial vegetative propagation. Did you know that gardeners and farmers use artificial methods of vegetative propagation to increase the stock of a plant? The first method is by cuttings. In this method, cuttings may be taken mainly from the stem or roots of parent plant. Did you know that 5 months before I have grown these rose plants? I had only one cutting and now I have two plants which are growing successfully. I had taken the cutting from a parent plant and placed in a suitable soil and under right conditions, and the right conditions were sufficient nutrients, water and sunlight. And now these plants have developed their roots and shoots systems. This one was parent plant and later I planted this cutting and this one is new plant. These cuttings must have a meristematic region from which growth can occur. Roots and shoots grow and develop into a plant identical to the parent plant from which the cuttings were taken. Initially, the cuttings were taken from this plant and later on, now I have a new plant that is identical to its parent plant. These roses are grown through cuttings. Now I have a sweet potato to show you, and which is an enlarged root. Farmers place it in moist sand or soil until it produces several plantlets. Then the plantlets are removed and planted. And now I am showing you my grapevine, which is a parent plant. On 14 February 2020, I took this cutting from that parent plant. This cutting had meristematic region from which now growth is occurring. And now this cutting is growing successfully here in this soil. This process is used to produce many plants from a single plant. Unfortunately, this cutting is not successful here. Dislodging occurred in this cutting and the growth has been stopped. Now I will discuss about grafting. Do you know that this plum tree has a well developed root system? 5 months before, I took the stem from parent plant and made a cut and attached with this parent plant. I wrapped that stem with a polythene bag and covered it with scotch tape. Now this stem is growing successfully here. Now this parent plant is serving as host tree. In grafting, a piece of stem is cut from the plant and is attached with another plant with well-established root system. After a while, the vascular bundles of the attached stem piece and the host plant are connected to each other. The stem piece and the plant begin to grow together. This method is used to propagate many roses, plum trees, peach trees, and various seedless fruits, for example, grapes. The benefit of this artificial propagation is that the new plants are genetically identical to its parent plant. The offsprings produced through vegetative propagation are genetically identical, therefore, beneficial characteristics can be preserved. In vegetative propagation, there is no need of any mechanism of pollination. It helps to increase the number of plants at a rapid rate. I have discussed earlier that I had only one cuttings, and now I have two well-developed plants. The organs of vegetative propagation enable many plants to pass over unfavorable conditions. Plants bearing seedless fruit can be grown only by vegetative propagation. For example, oranges. And now I have a lemon to show you that this lemon is grown at farmhouses through artificial propagation. If I will take the seeds and grow these seeds into soil, then these seeds will not grow, because the farmer that was at farmhouse used the artificial vegetative propagation to get enough production of lemon fruit from its tree. And now I will discuss about the disadvantages. The plants do not have genetic variations. Some species specific diseases can attack, and this can result in the destruction of an entire crop. Next one is tissue culture and cloning. This is the method of propagation which is also called micropropagation, since it uses only a small part of a plant. So here you can watch that tissue pieces from the parent plant are used, then tissue placed in suitable medium here, then tissues developed into small plants and new plants sown to grow. These are small plants and these are the new plants from these tissue pieces that were taken from parent plant. So this is tissue culture, cloning, or micropropagation. This is the latest method of vegetative propagation, and in this method identical offsprings are produced from a single parent using its vegetative tissue or cell. Tissue culture is the technique applied in this method. Tissues are taken from any part of the plant and are put into a suitable nutrient medium. This medium has nutrients. The tissue cells start mitosis and produce mass of cells called calyces. And these calyces are transferred to other medium that contains different hormones for the formation of roots, stem, and leaves. Calyces make these structures and grow into new small plants. The small plants are then planted in pots and then in field. So that was all about artificial vegetative propagation, cuttings, graftings, advantages and disadvantages of artificial vegetative propagation, and tissue culture. I hope this lecture was a good source of information. Thank you and have a nice day.